Hello everyone. My name is Jed Brown. I'm a PhD student from the University of Sydney, and it's a pleasure to be speaking to you today about some research we conducted in 2021 on soil seed banks. We were looking at the plant richness and diversity and seed density in the soil seed bank. We're also looking at the above ground plant species richness and diversity. And we were looking at the relationship of these biodiversity metrics with a non-native invasive C4 perennial tussock grass, bit of a mouthful, Eragostis curbula, more commonly known as African lovegrass or weeping lovegrass. So I'll break down a little bit of our biodiversity metrics and what a soil seed bank is. A soil seed bank is a reserve of seeds in the soil that are a reflection of the past and present species composition above ground. You can think of it like the memory of the species composition of plants that were in the ecosystem or are in the ecosystem. Species richness, probably all familiar with species richness. It's a metric that just measures the number of species in a given area. So if you have a 1 by 1 metre quadrat or square, if you have 10 species in there, you can say your species richness is 10. Species diversity, a little bit more complex. There you're looking at the number of species in an area, and you're also looking at the count of each one of those species, and you can do different formulas to get a metric for diversity. For our research, we use Shannon Diversity Index, has a particular formula that goes with it. Don't need to get bogged down in that at all. But yeah, so those are some of the metrics we were dealing with in this research. The star in question is African lovegrass. This is a non-native invasive, so it's probably important to note that this research was conducted in Australia, so it's a non-native invasive in Australia. African lovegrass is spread from its original area, which is in southern Africa, to many continents throughout the world, in the US, Argentina, Japan. People have different perspectives on the species, but in an Australian context, for the majority, it is considered a non-native invasive. It's a C4 grass, so that's referring to its photosynthetic pathway, opposed to a C3 grass. C4 grasses tend to produce more biomass, and they tend to be good at handling drought, so they can handle dry conditions pretty well. Opposed to C3 grasses, and that's in general terms. There's of course exceptions. It's perennial, so it's going to grow and seed over multiple years, and it's a tussock grass, and that's just going to refer to its growth habit. So if we have a look, we've got there. This top image here, we've got a African lovegrass tussock. It's kind of solitary at the moment in this image. It's growing in sandy soil. It's pretty dry. It's got some adaptive traits to handle the dryness with its curling leaves, reducing surface area. So yeah, pretty hardy little grass. And in this lower image here, you can see African lovegrass in more of a monoculture. It's kind of taken over the system, and it's really restricting growth of any other species there. So it was originally introduced into Australia for soil stabilisation, and it was an auxiliary hope that could also be used as a potential pasture grass for grazing livestock. Some of African lovegrass traits are high seed germination rate, high biomass production, a deep root system, and the ability to produce thousands of seeds per plant. If it was a palatable pasture grass, it would be amazing because it's spread, it's self-spreading, produces a lot of biomass, but as it turns out, it's fairly unpalatable for most of the year for livestock. There's a few exceptions of that. So due to these traits, it's going to act as an agricultural and environmental weed. Environmental weed in that it seems to be decreasing the richness and diversity of other species. So we've got African lovegrass, seems to be a problem, both agriculturally and environmentally. So we want to find out some more about the species, really assess its impact, and then we can decide how much resources we're going to throw at this issue, or how much of an issue it is. So we've come up with some research questions to drive our research. We've got, what is the relationship between African lovegrass cover and the soil seed bank? That's kind of our driving research question. But we also have another one, which is what is the relationship between African lovegrass cover and above ground plant richness and diversity? So just take a minute to talk about where we were conducting our research. We conducted our research in a region called the Snowy Monaro region. So this is a political boundary, but it also loosely aligns with a more physiogeological region called the Monaro Tablelands. This is defined as an undulating land plain with tabular basalt reliefs, granite tors, and these break down into grasslands with, talking in very general terms here, basalt and granite-derived soils. Of course, geology is a little bit more complex than that, but that's a nice general overview of the region. In an Australian context, this region is fairly high altitude. Australia doesn't have lots of large mountains. So we've got an elevation range of about 400 to 1,500 metres above sea level. The region's also home to Australia's highest peak in the form of Mount Kosciuszko, which is over to the west a bit more in this area here. We have average rainfalls of about 780 millimetres per year. That is brought up a lot by the Great Dividing Range, which gets a bit more rain than the surrounding, or the lower tablelands here, which are predominantly grasslands. So, experimental design. We had a pool of about 20 or so properties. These are farms, which we could have chosen. And based on a certain selection criteria, we narrowed that down to 8 different sites. So these sites are essentially just farms of varying use. The selection criteria was based on density of African lovegrass infestation on the property. If we had, we had sites that had basically very little or no African lovegrass, and we had sites that had very high density African lovegrass. So we wanted to have a bit of a gradient going from low to high. In each one of our sites, we chose 3 subsites. So we've got an example of that here. This red box is an actual site. It's a farm. And then within the site, we had 3 of these subsites. Within each subsite, we had 10 quadrats, so squares, sampling squares, and in each one of these quadrats, we would take a soil sample, we would take above ground species richness, and above ground species diversity, which we were measuring with cover percent. If we do the maths, we have 8 different sites, 3 subsites within each site, and 10 quadrats within each subsite. We had 240 sampling points. Here we have just some images of what we were doing. So square here, we have a quadrat, which has basically 100% African lovegrass. With the soil collection, we collected soil to be used in the soil seed bank study. We basically drew a grid within this quadrat, and at the intersecting lines, we collected soil and we amalgamated that, and that was used for our actual soil seed bank study here in a temperature-controlled glasshouse. This image here, we have a quadrat in more of an uninvaded system. So yeah, this is kind of an example of the gradient. We had no lovegrass, all the way up to very high amount of lovegrass. Results. So what we found was the above ground species richness and diversity seemed to be negatively correlated with proportional cover of African lovegrass. This kind of makes sense. As we have more lovegrass occupying a quadrat, you expect to see less species being present in that quadrat. We were using a generalized linear mixed effects model to assess this data. We used Akaki information criteria, AIC here, to assess model fit. The full model had a predictor variable of African lovegrass cover and a response variable of either richness or diversity. And in both cases, we have the full model having a lower AIC, meaning that it's the better model in this case. So here's a plot to kind of try to explain the data a little bit. It's a little bit confusing with all these lines. So we were using a generalized linear mixed effects model. We had a random effect of subsite. So each one of these thinner lines represents the subsite, and this thicker line here is kind of the general trend downwards. So these are the kind of the regression lines, and each one of these points is the observed data, and the numbers correspond also to a subsite. As we can see, we've got proportional Eragossus curvula cover on the x-axis. As proportional Eragossus curvula increased, we saw a decrease in above ground species richness. Similar to the previous slide, we have log Shannon diversity +1. We needed to do that just for meeting a model's assumptions, but the general trend is the same, if not a little bit more pronounced here with diversity. Along the x-axis, we have proportional Eragossus curvula cover. As that increases, we see a decrease in diversity. So as for the soil seed bank component, or the research question around the soil seed bank, we found that regardless of African livegrass proportional cover, below ground species richness and diversity were not affected. Here's an example of some of the soil seed bank diversity we saw. So we've got a nice range of grasses and forbs. But that's really just the tip of the iceberg of this research. You may have noticed that with our soil seed bank species richness and diversity, we really didn't split it up into functional groups, which is kind of important. Lumping that all into one big bucket may not be the best way to assess the actual effect of African lovegrass on the soil seed bank. So for the paper presented here at this conference, that's the data we've presented. But since then, we've analysed the data a bit further, and I just wanted to quickly kind of go into what we've found since we're doing that. So we came up with a new research question to look at the functional diversity in the soil seed bank, and we've split it up into native and non-native species in the soil seed bank. And we've got this new question is, what is the relationship between African lovegrass and the functional groups in the soil seed bank? We found that African lovegrass cover increases. As this increases, as African lovegrass cover increases, we see a decrease in native seedling density. So breaking that down, if we have more of African lovegrass in the system, then we're going to have a decrease in native seedling density, less natives in the soil seed bank. That's not great if you're looking at a restoration effort. If you've got an area that's full of African lovegrass and you want to try to rehabilitate, if you do happen to manage the above ground biomass, you may need to add natives or desirable species into your system to try to get it to a desirable state. African lovegrass presence seems to decrease native richness and native cover density. So it just being present in the system seems to have a negative impact on the natives. African lovegrass or Eragossus curvula presence seems to increase above ground non-native richness, non-native cover density, and non-native seedling density. So not only are we seeing less natives in the soil seed bank, or at least in terms of seedling density, we're seeing more non-native seedling density. So, a bit of an issue there in terms of if we want to restore a site to a pre-invaded state. So how do these findings relate to other research? There's a review done called The Legacy of Plant Invasions, Changes in Soil Seed Banks of Invaded Plant Communities. Pretty relevant. And they've summed up some findings from a range of different research. They suggest that changes in the soil seed bank may be a symptom of land degradation. In our case, each one of our sites has some history of livestock management. Now, not all livestock management leads to land degradation, but you can get situations of overgrazing, soil compaction, and that can open up space for invasive species to take advantage of. We found a similar conclusion that impacts of invasive species is greater on the soil seed bank density than on the soil seed bank species richness and diversity. We came to pretty much the same conclusion, so there may be some generalised trends there. And finally, a solid understanding of the legacy of plant invasions on soil seed banks is still lacking. I think we've made some progress, but there's still a lot of unanswered questions. So yeah, more research to be done is the takeaway there. That's all I have. Any questions, feedback, you can scan that QR code. That'll take you to a Google Forms. If you want to give me some positive or constructive feedback, that'd be great. Otherwise, my email's there. And yeah, thank you to the A.W. Howard Memorial Trust and IGC for allowing me to speak today. Thank you.
Mr. Jay, before we go to questions, I'd just like to know if our next speaker, Lou Bai, is present or anybody making the presentation? No? So that means we have a we'll have a 15-minute question and answer period and discussion.
15 minutes, sure. Hey.
I'm curious, and I'm sorry I came in a little bit late. Do you have an
Yeah. Yes, please.
I'm sorry if I missed it. I came in a little bit late. Do you have an idea about the residence time of African lovegrass on your sites? Any idea of how long it's been on those sites?
Sure. Yeah, so it ranges. The species itself was intentionally introduced into Australia around about 1930. That's when it was originally, brought into the country. Each site varied. Some sites, the land managers believe it's been there for like 50 years. Some sites, it's only very recently been on there. So that's definitely a factor in the study. But yeah, it varies between species. Something we could definitely dig into more and find out some more interesting things there. But yeah. Yeah, definitely. Hey. So the question was, do we measure the seed bank in the dung of animals?
Yeah.
We didn't, not quite within the scope of our research with limitations in funding and resources. But again, yeah, an interesting avenue. And I haven't seen any research on African lovegrass in particular making it through the digestive tract of livestock. But yeah, definitely something that would be interesting to find out. Thank you.
Just to let you know, the second speaker is not here, and so we're continuing our discussion, just to let you know. I do have a question also.
Yeah, go for it.
When you collected the soil, what depth were you collecting?
Yeah.
10 centimeters?
Yeah, so it was about 8 centimeters. We were just getting the topsoil. We were just using a hand auger. The diameter was about 5 centimeters as well. And then, yeah, we would amalgamate that into a single sample, and that was basically what we put in the soil propagation trays, and then we would identify things as they popped up. Yeah.
Very good. And could you explain how you identified them and how you became an expert in identification?
It's a pretty laborious process, not going to lie. As the species popped up, it's very hard to identify seedlings. I don't know if anyone's done that before. So what we did was we had just a large number of different pots. When they popped up, we let them grow for a little bit so we could kind of take them out of their propagation tray, put them into something else to grow, and then, cross our fingers, hopefully they survive and get to a point where we can get an accurate ID. But yeah, it's a slow process, it's a patient process, and yeah, lots of learning along the way.
Sorry, I'm going to ask another.
Yeah, go for it.
Did you or are you thinking about looking at the similarity between your seed bank diversity and your above-ground plant?
Yes.
Could you please repeat?
Sorry, the question was, did I look at the relationship between above ground diversity and below ground diversity? Sorry for paraphrasing, but yeah. Yes, it's in the works to answer that question. I wouldn't say that I'm confident in being able to answer that to satisfactory level. But yes, we are looking at it, and yeah, we're not quite there to be able to talk about it in terms of confidence in my own way to, yeah. Good question. Yes, look out for it. Hopefully I'll publish that soon.
Other questions? I've got another question for you then.
Yeah, go for it.
You described the results as the non-native diversity and richness also occurred. Was that only the lovegrass, or was it you identified other non-natives?
So lovegrass definitely contributed to that because it was counted. It was involved, yeah. But yeah, we were seeing a lot more non-natives. That may be a case of the non-natives being found are like just other pasture grasses that have been introduced to try to improve the quality of the pasture. But yeah, there was African lovegrass was included in that count. So that does make sense that if it's there, it's going to increase the number of non-natives in the site. Cool.
Well, don't run away, okay? Because you get the opportunity to well, you haven't defended yet your thesis.
That's true. No, I have not.
Okay,
No.
so this is an opportunity for us to help you prepare. No?
Thank you. Yeah, no, I appreciate it.
Okay, very good. Yes.
Thank you. So, as my understanding, it could be a bigger biomass, higher biomass than the native plants in Australia. So, is it it's because the Australian grassland, the lands is usually having some problems, right? Salt. So, how is it performing under the salty conditions, salt tolerance? Have you measured that?
Salt. I might have to play ignorance to that one. I'm not quite sure how it handles salt. It's hardy. Let's put it that way. It grows pretty much anywhere. So just intuitively, I think it would be fairly salt tolerant. It can handle acidity pretty well, which I know that's not a question you asked, but it can handle acidity fairly well. And in terms of biomass, yes, it does produce a lot of biomass. What we found, particularly during drought, it manages to retain its biomass a lot more to the natives as well, which isn't great. I also conducted this research during the bushfires we had in 2020, the Black Summer bushfires, and yeah, the lovegrass definitely retained its biomass, and talking a little bit anecdotally here, but it seems that the fires that went through the African lovegrass sites, they burnt quite hot, which isn't great in terms of changing these disturbance regimes. So, yeah, sorry, I kind of went on a tangent there, but you mentioned biomass, so I thought I'd shoehorn that in. But yeah, sorry, in terms of salt, I'm not too sure. I might need to go do some reading about that.
Okay, I'll ask another question.
Go for it.
About when you had the quadrants, it looked like they were somewhat distributed, but not totally equally distant from each other. How did you decide where to put the quadrant?
Good question. I like that question. So I used a GIS software to randomize it for me. So I had predefined sub-site locations, and then using, I'm fairly sure I just used QGIS, I set a threshold boundary that they can't be more than, I think it was about 30 meters from each other. And then it randomly distributed 10 points within that sub-site. So it is as random as I could make it, and then I would go out with the GPS, find the location of where the GIS told me where to go, and then that's where I did. Some sites, some locations may not have been ideal, may have been like in a riverway or something like that, so I would just kind of the nearest, suitable point. Yeah.
I'll play.
Yeah.
Poor guy. So you presented proportional cover data
Yep.
for your regressions. Did you also look at absolute cover, and did you see a similar pattern?
Yeah, we did. Similar pattern. I think we just went with proportional. This may not be the most scientific answer, but I'm fairly sure we just picked one or the other, and we just went with proportional. But I think there was a similar trend the other way around.
Okay, other questions?
I'm comfortable up here now, so.
Okay, well, that's right, you're doing really good, thank you so much. And what about tiller density? When there's because it's more of like a tussock, right?
Yep, a bunch grass or a heap grass,
so
yeah.
It competes that way, right? I mean, it's occupying space that isn't necessarily occupying tiller space. Or expound on that.
Yeah, I think its competitive ability, I think, stems, excuse the pun, from its sheer biomass. It just produces so much, quite a lot of tillers, and just occupies the space. It's dense. It also produces a lot of litter. So we have another portion of our research where we were looking at litter production as well, and yeah, it just produces so much more litter than the natives as well, and I think that creates this sort of restrictive boundary on the surface, and that prevents germination of other species.
Okay, very good. I'm going to follow up in indicating, like, so did you actually measure residual biomass?
Like
on the surface?
Like litter biomass?
Yeah.
Not in this study, but in another bit of research, part of my PhD, we did, yeah.
Okay. And in 1 square meter, did you occasionally have just single plant that you identified as one single plant?
Yeah, it's hard, just because of the way that it grows. It kind of grows in these rings, and it'll die off in the middle.
Is it one plant or multiple?
It's one plant. It's one particular plant when it grows as a bunch. Yeah. That's one African livegrass tussock. But in terms of in a meter squared, we often got 2 or 3 actual tussocks. So they grow quite close to each other.
We have time for another couple of questions if you want. And Jed is being very, very productive up here.
Great.
Well,
I might take
thank
my
you so
leave if
much,
that's
Jed.
okay.
Okay, yes, sir.
Thank you.
Absolutely. Thank you. Martin, this is your presentation, yes? Okay, very good.
Do you need to go to the first slide?
Oh, I see.
Yeah.
There you go. You got it under control? Okay. All right, very good. Let me grab something here. Okay, our next speaker is Martin Kaminde.
Yes, that's right.
Okay, very good,
Yeah.
from Germany, and he's got a 15-minute presentation, but presentation number 4, I don't know, it is Min Zhao present, or anyone from Min Zhao's group? So Martin, if you want to avoid the long questioning period, you can extend your presentation as long, up to 30 minutes as possible. Thank you.
Okay, thank you, Alan. Hello, my name is Martin. I'm from Germany, and I would like to talk about below ground productivity. I will try to slow down the speed of speaking, and the topic you see it over there is below ground productivity in patches of heterogeneous grass swards after nearly 2 decades of low intensity cattle grazing. Please don't fall asleep while reading that. The topic is simpler than the title. Okay, to start with, initially we asked, does stocking intensity affect soil carbon? This is our background. And we had a look into the literature, I had a look into the literature. It's not so compelling evidence for at least temperate or continental grassland if you look into experiments that are really replicated and also long-term. There is some studies that show increases, others show decreases. Mostly differences are not significant. You see it displayed in this table. The red ones are significant changes in soil carbon related to intensive grazing. But especially in continuous grazing, there are some patterns that we might miss and that we should consider in future studies. And one thing is that with lower intensity, at least in temperate grassland, we have higher variability among pastures, but also especially within pastures. Johannes Isselstein was talking about that the other day already, but if you see an extensive low input grassland system, it's highly diverse in many terms with respect to botanical composition and also the variability of the grass sward composition. And what we see that under low input, continued grazing, especially under patch grazing, that the diversity in the heterogeneity could cause some differences that might explain the lack or the presence of differences in soil carbon. And when we refer to patch grazing and continuous stocking management, this is what we see here on the picture. You have either short areas adjacent to taller areas. And these areas that are short, they are frequently and often defoliated all the time. So there is nutrient export from that area. And that means that soil nutrients also deplete in relative terms. In essence, the diversity in those areas can be higher, and also changes in soil water and soil temperature might occur. Whereas on the other hand, adjacent to those short areas, you have the tall patches. And these areas, at least under continuous stocking, they are hardly ever defoliated. So there is no nutrient export. It's rather an increase in soil nutrients because of the droppings of the animals. They drop randomly. So if you don't have an export but a random dropping, it will increase in essence, and that means that diversity within patches differs, and also among patches differs particularly. And also probably light competition plays a role here. But just to sum up, we know from previous studies that those patches, they are very stable over time, and that the proportions particularly depend on the stocking intensity. If you do it more intensive, you have more shorter areas. If you do it more extensive, you have more taller areas on a paddock or on a grazing land or on a rangeland, whatever, yeah, in the landscape. Now, coming back to the carbon topic, if you think of soil carbon as a balancing approach, it depends on the input and on the output of carbon into the system. And roots are key here, not because of the simple root carbon input, but especially from the root turnover and also from the decomposition and the rhizodeposition, which is 2.5 point the magnitude of root production. If you think of patches now, you might see a trade-off. In the short areas, we see the continuous defoliation and export of carbon. That means the plants need to invest above ground all the time, produce leaf. On the taller areas, you don't have this effect, so plants can invest below ground whenever they like. So this trade-off might cause that there is difference in soil carbon related to those patches, and this is what we are analyzing in this ongoing research project. This is mainly the idea. So the roots are key to carbon input into the soil. And the particular question would be, what if soil carbon stocks differ among patches within pastures? That would make soil inventories really unreliable. So we look into that particularly. There is a part of the sentence in the hypothesis written in black. This is important for today. The other part is not important. So the main hypothesis is within pasture variability of below ground. So we look into the below ground biomass production, and we look particularly only into the roots, is larger than the variability between pastures that are grazed at different stocking intensity. So we only look into the pasture environment, into one pasture environment, and differences here. And the procedure is that we use a long-term research platform located in central Germany. You see it here. It's the 4-biobean continuous grazing experiment. It's now 2 decades old or more. The grassland is even older. And it's 3 grazing intensities, in essence, with 3 replicates in 1 hectare paddocks. 1 hectare size is each paddock, so we have 9 paddocks, and 1 hectare is the average size of grassland in Germany. So it's really representative, okay? There's only grazing, there's no cutting, and there's no fertilization for at least 30 years. So what we see there is the product or the consequence of grazing. And from a stocking intensity gradient, we're somewhere in the middle, but more or less still in an extensive system. We do take 2 treatments out of this experiment, which has 3 treatments, and they are called moderate and lenient intensity. They are managed according to sward heights. The one is on average 12 centimeter. The other treatment is on average 6 centimeter. And probably around 400 versus 200 livestock unit grazing days. On average, a stocking rate of 1.1 versus 0.7 livestock units per hectare. So it's extensive, yeah? And the variability within those pastures is huge. The grassland itself is quite diverse. It has 70 plant species recorded over the whole experimental area. It contains 11 species per quarter square meter, so it's semi-diverse. And approach we use is in factorial. It's a factorial design in the end, spread over the experimental area. I don't know if you see. You see. So we use the stocking intensity treatments. We know that those patches are stable over a very long time period, so we exclude specific areas, and then we install our measurements into tall and into short areas. This is our first factor, the patch. Then the second factor is the botanical composition, because we want to make inference based on the effect of potential effect of botanical composition. So we stratify for grass-based, grass-dominated areas versus dicot-dominated areas to install our equipment. And we also know from animal movement data that there are areas which are more enriched in soil nutrients and others that are more depleted in soil nutrients. So this is another factor we look. I think you probably know that soil nutrients affect root production, therefore we look into that too. We have 3 replicates, in the end 48 observation plots, which you see here displayed, and what we do is in each of those exclusion zones, so we exclude the grazing animal from that areas, we installed mini rhizotrones tubes to 30 centimeter depths, and this is a nice system in order to capture a lot of data in very short times, but you don't have the mass. You only have the root length and root length density. Therefore, we use the ingrowth core technique to relate the root length and the root mass. And we do that dynamically, so over the whole growing season in a 2, 4, and 8 week interval. So very regularly. It's a lot of labor. And then above ground productivity is measured based on sward heights, compressed sward heights, and we link that to manual clipping so we know what's growing there. We have some covariates, plant water, plant available soil water, and temperature. And this is how it looks like. So on the left side, you see the below ground net primary productivity measurements based on ingrowth cores. I don't know if you're familiar with ingrowth cores. Since we have time, I could explain that too. So it's a mesh bag you insert into the soil into a specific depth, and you fill that mesh bag with soil of the site. The soil has to be root-free, and the amount of soil you fill into the mesh bag should match the surrounding soil bulk density. So you correct for that first. And then you let roots grow into those mesh bags and recover those mesh bags after 4 or 2 weeks or 8 weeks. That depends on your approach. There are studies doing it once per year, others doing it weekly. And this is how you basically measure gross root production if you are not too long to get problems with root turnover. So let's say it's a below ground net primary productivity here. And the other method we use for manual determination is the root auger method, also a very laborious method, which we use to assess standing root mass and to 30 centimeter soil depths in regular manners, and we link that data also to soil carbon to check if there is a relationship between root mass and soil carbon. And yeah, that's basically the methods. Now I'm coming to the results, and since it's ongoing, it's only preliminary, and that is the display of weather data from last year. It was very dry, 40% less rain, and pretty hot, so plus 1 degree Celsius last year, which is getting more and more common, but yeah, it affected, of course, what we measure. You see it displayed in the picture. It was pretty dry in August. A lot of senescence and litter built up. Yeah. However, the results of above and below ground productivity look like that. You see on the left, ANPP, it's for above ground productivity, and BNPP for the below productivity. And on a daily basis, measured per square meter over several months. And we would have expected that tall patches grow better. But that was not the case, at least for the last year. We had no significant effect, only for the months. For the below ground productivity, there was a patch effect in interaction with the months. Yeah, we had higher productivity, at least sometimes in the year. In June, I think, you see that here. The productivity in tall patches was larger below ground. However, in total, if you total that, it was the same. Yeah, but there seems to be some variability, and now we need to start to think about what's happening there. And one issue with respect to the carbon investment is, of course, okay, in short patches, the plants need to invest above ground in order to survive after defoliation. And in tall patches, it could be that the light competition also triggers the plants to grow above ground, and they don't have carbohydrates in excess to invest in below ground productivity. So probably light and interaction with light and also defoliation intensity plays a role here. We need to go into that deeper. When we measure light on a monthly basis using AccuPar equipment, it's LRE measurement technique. I don't know, probably you are familiar with that. Okay, so the question is, with respect to the variability within pastures, yeah, there is some. What does it look like among pastures? If you manage them long term under varied stocking intensity, and you look into the standing root mass, so there's no difference. Yeah? We did also find a seasonal effect here in the live root production of biomass, but not among patches and also not among grazing intensities. So it doesn't matter how extensive you graze. Okay, and this would be my conclusion based on the hypothesis from the beginning. So there is some variability among patches, but we need to go deeper, further. And the initial starting point was the soil carbon stock, of course, and this is the first data of carbon stocks calculated based on manual sampling of soil and to 30 centimeter soil depth. And we don't see any relationship between patches and soil carbon stocks in the soil. At least it's not significant. Yeah. And with that, I think I like to stop and thank you, the funding agency, and all the people who are related with this work. Yeah. Thank you.
Thank you, Martin. I want to make you don't go away, please. All right, so our next presenter apparently is not here. And just to let you know that the time is up for 15 minutes, and very good timing. But we have time for asking more questions during this 15 minutes or other discussion that you might want to have. So is there any question for Martin based on the presentation? There's a question there.
Where do I go? Hello, very interesting presentation. Thank you for that. I would like to know a little bit more about the soils you were having, because you were having so many different species all around, and more related to the soil physics characteristics like permanent wilting pointing and whatnot. Was that changing much, or it was more general that you were using?
The soil physics, you mean, or the soil chemistry?
Physics, please.
The soil physics. Okay, we do not check that. We looked into soil bulk density over time, and also between those patches I mentioned. Yeah, and there is no change. You would expect that in areas that are used frequently, there's more compaction, and less in those that are not so often visited. But it's not the case. It's not significant. Yeah, so from this respect, it's the same. And also the texture in this 9-hectare pasture is pretty much the same everywhere. Doesn't vary that much. And there is quite some variability only with respect to soil nutrients and also micronutrients, as based on previous research, not mine. And I think I didn't mention it, but it's a silty loam soil. So it's high, with a pH of about 6. So it's overall a good condition to study that. But it's not that deep. It's 30 to 60 centimeter deep. Yeah. But that's all I have on soil physics.
Is this all right? I just had a question regarding whether you looked at the variability in soil water holding content throughout your pasture, and whether there was a relationship between soil dryness and root growth.
The soil hydraulic conductivity, you said?
Water holding capacity, or
holding
I guess
capacity.
hydraulic conductivity is related.
Yeah, I just checked the. We use volumetric water data, and I think, if I'm not wrong, yeah, it differed among depths, obviously, but not among our treatments. We stratified for, no, so not among the patches, and also not among in relation to the botanical composition or stocking intensity. It's all the same. Yeah, you have to really consider this. The system is running on 4 cows per hectare versus 3 cows, or 3 versus 2 cows per hectare. This is the basis, the stocking rate. Yeah.
I have a small question. Is this an ongoing project, and we will repeat the measurements, and maybe some results will change?
Yeah, maybe yes. Probably the results will change. It's this particular project I'm running now is a 2-year project. We are now in the 2nd year. People are probably outside at the moment sampling, and so for the below ground productivity, we will have more data. We have already data from 2018 on the changes on the trajectory of carbon in this pasture, and the earliest data was from, I think, 3 years after starting this experiment, and there is an increase over time in soil carbon as it accumulates in grassland soils. This is pretty common, but it does not depend on the stocking intensity over time. Yeah, but at least from carbon content perspective, but also from the stock, since the bulk density didn't change. Yeah, so there will be more data.
Thanks for the presentation, Martin. It was really interesting. There was no difference between the stocking intensities, but they were moderate and extensive, but they were quite similar. 0.4 of a difference in the livestock units overall. Do you think you get a much higher level of carbon sequestration if you really dropped it to maybe 0.5, or if you increased it dramatically to 2.5 or 3?
Thank you for the question, Kieran. Don't know if you see that. These are all three treatments over there. In the upper figure, you see red, blue, and gray. It's 0.3, 0.7, and 1.1 livestock units per hectare, and it's really the same over time. It didn't change. That also means, of course, you could go for more intensive grazing, more intensive food production. You don't have any trade-off for soil carbon. but we didn't balance that against methane. Yeah, with 3 cows per hectare, you have more methane per hectare than with 2 cows, of course. However, I don't know. I need to balance that. If the increase in soil carbon captures the additional release of methane, so the context.
Obviously, I know this works, so it's more a comment than a question. And maybe a question to the audience who are familiar with this type of research. There is quite a debate in Europe as to how grasslands can store carbon as a mitigation strategy to climate change. Well, our preliminary conclusion, as Martin was saying, is obviously this is old permanent grassland, not having been plowed up for we don't even know, decades, obviously, and it seems as if given this situation, the potential to store more carbon in those soils, in those mineral soils, is obviously very limited, and you could manage as you like. It would probably not make such a difference if you compare with the situation where you have, we have quite a bit of data around with temperate grasslands or grasslands being established and then you have a crop stand for 10 years or so, then you may see quite big differences in how you manage the grasslands. But this is obviously not the case if you take permanent. So as a conclusion, a preliminary conclusion we have is it's if you think of grasslands being a potential to mitigate climate change and to help carbon storage in the soil, you must not plow up the permanent grassland. This is the first thing we would think as a conclusion. But maybe someone can comment on that.
Thank you. Mainly related to what you say, up to which level we can go in terms of intensification. We have some long-term studies in Brazil as well. Extensive pastures, pretty related, 0.5, 0.8 units of animals per hectare. Different levels, then we start applying a little bit of fertilization, then higher amount of nitrogen application up to irrigation. And also we have some systems that we integrate with forest, right? So we found that when we get up to the irrigation level of intensification, we have we have decline of soil stock, carbon stock. So we don't quite know exactly what is the mechanism that is happening, what is going on in the soil that gave us this kind of result. And for us, higher amounts of nitrogen not going up to irrigation would free us some area to have forests, for example. We have something called land saving effect, and we saw that from a 3.7 hectares area of extensive production, you can produce the same in 1 hectare of intensive production, and plant 2.7 hectares of forest in the rest of your area.
Yeah.
So, yeah.
Thank you.
Other questions? We've got some time.
So I have a quick question about your root measuring methods, as I'm not very familiar with it at all. That's really neat stuff. But you said you did some exclusion in it. Was it to protect the equipment? And if so, how do you make sure that you're tracking, you're actually reading those differences in treatments?
Yeah, it's important for the system because the mini rhizotron, it's a plastic tube that comes up out of the soil. If you have trampling damage, you're gone for 1.5 year more, because you have to install it 1 year before you start reading the first mini rhizotrons. Yeah, so if that is trampled by the cow, it's lost. Therefore, we need to exclude, and also the ingrowth course probably, and we make sure to maintain a short area by manual clipping. So we imitate the grazing. Yeah, that's that's the only possibility.
Okay, do you see any potential way of because I feel with some of those, I don't know for sure necessarily, but I feel in some of those ways you might miss out on some of the the manure deposition or that the potential effects that that might have on soil organic carbon stocks. Do you know of any methods, or like, is there a potential future for methods that will let you measure these things while grazing is happening at the same time?
Yeah, probably. There are also mini rhizotrones that you can walk in forests, for instance. So really huge equipment, and maybe that's something that you could install below your pasture with really huge investment. But so far, I think for our purpose, it needs to be it must be some more artificial kind of, and there is also those methods you use with scanning devices. There are labs, root labs, and also more, I think, computer tomography is coming in also, but these are more based on taking a soil column into the lab. Yeah? We are still outside under real conditions, and that's, I think, the advantage of using those methods.
Okay. Right. Excellent. Thank you.
There's a question back there. May I interject just a little bit in that you said that there was a 20-year grazing experiment, and then only for the year of your measurements, or the previous year and year, then you exclude.
Yes. Yeah.
There's still a legacy effect.
Yeah. Yeah, you're right. Thank you.
Yep, my question was also about the methods you're using, so I'm not that familiar as well. So could you comment on, I guess, what your methods you're using and what data you're getting out of them? So I think you mentioned the mini rhizotron, the mesh bag cores, and also the soil sampling and how they kind of work together.
Yes, the mini rhizotron gives you a good estimate of root growth, actual root growth based on picture analysis. Yeah, you measure regularly, and you get the changes in invisible white roots in the channel and in the tube. So that's what you assess: changes in length, changes in length density, and maybe some other traits, fine root and thick roots. You can measure them, but you don't have actual mass. So we would link that data from the mini rhizotron to the ingrowth core, because we measure at same depths, and we capture then changes over time based on length changes, and also with the ingrowth core based on mass changes. So we could link that. So that's a pretty nice advantage to have actual root samples also for analysis in the lab. You need that probably, I would say, at least for some time periods. And then the auger method, it gives you an estimate of the standing root mass and the changes, essentially the result of decomposition and build-up of roots. So with our approach, the ingrowth core, you measure always the production of roots, but it can be that the decomposition at those times is much higher than the production of roots, and that gives you, and the auger data gives you then a picture of that, the relationship between growth and decomposition. And you can also assess the composition of roots by using different time periods. Yeah, if you leave the ingrowth core for 4 weeks inside the soil and compare it with 2 times a 2-week interval, if there is composition, you will capture it in comparing those intervals together. At least it worked for for corn, sorry. And last year in our trials, it didn't work out with 8 and 4 weeks.
It's not a question.
Yeah.
It's an offer. We can discuss it later, but I can offer that I take some undisturbed circles and scan it in a micro CT, and then we can have a relook to the 3D root development.
Yeah, great that you're also in Germany. Yeah, thank you, Katrin.
Thank you so much.
Thank you.
Okay, I want to keep on schedule, and I want to know if Guomei Yin, or if that anybody from Guomei Yin's group is here. If not, we have Lan Li, who is going to present, and you get to have 30 minutes in your presentation. Well, in fact, you can have even more time if you really want to, because it will be the end of the session after that then. So let's see, we'll get it up.
Yes.
Is it a PowerPoint or a
Yes.
PDF?
PowerPoint. No?
I don't know. Is it your folder? No, I don't think so. Is it your folder?
Yes.
Yeah, it is. Okay, let's try it here then.
No? Okay.
I don't know.
Start. Okay.
Very good. Just X out of there. Yep, very good.
Okay.
Okay, you take it from there. Thank you so much.
Okay, thanks for your attention. Because she didn't get a visa, so Dr. Li Yuanshi, she cannot have the opportunity to show herself. And I will. It's my honor to share some about her PhD research work in my country. And I'm Lan Li, the postdoc in the Lanzhou University of China. And the topic is, the effects of grazing and rain reduction on the growth and reproductive of the Artemisia calliparis population. No?
So you can, I'm not sure if we have a remote here. Oh, it's here right here.
Okay.
And you use the right button.
Okay.
No?
Why?
There
Why?
it is. Okay, you got it.
Why no slash?
You can also use the arrow button.
Okay.
Arrow key. This one here would go forward. This here.
Okay.
Yeah.
And as we all know, the grazing is the most intensive activities around the world, and the world was getting warmer, and maybe the dry place will get more drier, and the wetter place will getting more wetter, and the precipitation variability will change so much. And so the grazing and the precipitation change will also affect the phenology and the morphology of plants. We all know that Artemisia was distributed around the world, and in China, the Artemisia capillaris was native distributed, and many places there are also have some innovations places. So, based on the described before, and we put forward a scientific question about how do Artemisia to respond to grazing management and the precipitation changes, and we put forward the hypothesis about that. If we have the grazing and the rainfall reduction, the treatment, and the Artemisia will get more smaller, and will have a lottery maybe growth. And our study area was located in the lowest plateau in China, maybe in the northwest of the China, and the rainfall was decreased over the 15 years, and the temperature was increased, and the experiment site was located in the Huanxian Grassland Agriculture Research Station of Lanzhou University, may be located in the northwest of the China. And the grassland type was maybe temporary topical steppe, and the dominant species was Artemisia lepidisa and the stipabagina, and the soil type was the loess soil, and the altitude was maybe between 1,000 and 1,500 and to 1,000 and 1,700. The annual average temperature is 8.3, but and the annual average precipitation was 217. Then we set the experiment design maybe in the 2080, and we set two treatments about climate change, about the drought. We use the shelter to reduce the rainfall, and another is grazing management. A detail was that we set 3 treatments about the rainfall reduction was 0 for the control, and rain reduction about 30% and rain reduction about 16%. And the grazing management was, we have we have 23 years rotational grazing. It's about shift grazing, and we use the heavy grazing intensities. It's maybe maybe 13 ships per hectare, and we calculate the stocking rate was 8.6 sheep per hectare. And another treatment SG was we stopped the rotational grazing after 18 years, and then the control is enclosure. So the result, we saw that the year and the grazing management and the rainfall reduction have also the significant effects on these plant trees. We management this plant function trees from 2019 and until to the 2021, and we saw that the above ground biomass was decreased because of the variability of the precipitations, especially during the growth seasons. And we saw heavy rotational grazing decrease the above ground biomass, while the rain reduction 13% increase above the above ground biomass. We think the moderate drought maybe stimulates the growth of the artemisias. And we also find that the heavy grazing and the rain reduction 13% reduce the plant height, and we found the stop grazing after the 18 years, the plant height did not recover. And the heavy rotational grazing and stop rotational grazing increase the canopy diameter of the artemisia. It means after the grazing and the drought, the plants will get transfer their longitude growth to the latitude growth. And we used the canopy diameter to plant height to show the growth strategies, and we found that rain reduction 16% increase the ratio of them, and maybe it's about that heavy drought might increase the latitude growth strategies. And we also measured the growing points of the artemisia. We found that the heavy rotational grazing and stop rotational grazing reduce the growing points of the artemisia. And the reproductive bunch is also follow this, and we found that the heavy grazing and all the maybe all the grazing management reduce their generation potentials. And we use the reproductive branch to growing points to show the reproductive strategies. And we found that rain reduction 16% reduce the ratio of them. And it means the drought may decrease the sexual generation of the artemisia and transfer their anti-sexual to the sexual strategies. And we use the mean annual, maybe annual precipitation to show the precipitations, the relationship with between precipitation with growth and reproductive changes of the artemisia. We saw that with the annual precipitation increased, the plants will be getting transfer the longitude growth to the latitude growth, and and especially the growth season precipitation is a main driver of their growth and generation strategy changes. Then we draw the conclusion about that the drought made the growing effects more bigger effects on the populations of biomass of the artemisia, and the response strategy of artemisia populations and the grazing and rainfall reduction was longitude growth or the sexual reproduction. And under extreme drought, stop grazing is more conducive to the maintenance of the artemisia populations productivities, and under moderate drought, grazing can maintain the population's productivities. And I will acknowledgement about the IGC and our team and Lanzhou University and our stations. And if I, because it is not my familiar area, and if you ask questions, I can answer, and I will try to answer you, the questions. But if I do not why, and you can send an email to her, and I am sorry about that. Thank you.
Thank you, Longley. Any questions?
Maybe no question because, sorry. Thank you.
Thank you for your presentation. In relation to the growth strategies, do you have an idea why under a higher water restriction the plants were not trying to reproduce more? What would be I don't know, what would they be doing?
Oh, you mean the here? Yes?
Yes.
Yes. Why the strategy about did not change? Why the rain reduction did not change the reproductive?
I think it was one slide before, maybe. Yeah, no, but it's all right. Yeah. Okay. Thank you.
Thank you. I'm sorry about that.
That's all right.
Yes.
Well, one of the conclusions you made was that it changed from the increased drought and grazing pressure led to more reproductive versus vegetative.
Yes.
propagation, right?
Yes.
Is that related to your question?
Related to that.
Oh yes, and I have also some results because the rain reduction about 13% will increase the density of the artemisia, and the seedings will increase also. Yes.
A question: is Artemisia the sole plant that was investigated, and how was it in relationship to other forage plants there?
You mean to other species?
You only measured Artemisia.
Yes.
There were other forage plants there?
Yeah. Yes.
And so
Why? Why?
How did the other forage plants respond as well? Or was that not measured?
She had measured and some grass, herbage, and legume, and did not follow this trend, and another factors maybe. Yes.
Okay. I'm not so familiar with
Okay.
Others?